Welcome to this explainer. Today, we're going to completely flip the script on how you view viruses. Usually, when we hear the word virus, we immediately think of disease, right? We think of pathogens we desperately need to protect ourselves against. But today, we're shifting that perspective entirely. We're moving from seeing viruses just as disease-causing agents to understanding them as these vital, invisible engineers of our global climate. It's wild, but let's dive in, because what scientists are discovering right now is truly mind-blowing. I mean, it sounds completely counterintuitive, doesn't it? Can a virus actually help save the climate? But recent experiments in both our oceans and our soils are proving exactly that. These microscopic entities are nature's hidden regulators. They aren't just, passive particles floating around. They're actively and directly controlling how carbon is captured, processed, and stored on Earth. By the end of this explainer, you'll see exactly how the tiniest biological entities on the planet might just hold the key to understanding massive global carbon fluxes. To really wrap our heads around the sheer scale of their impact, consider this staggering statistic. Marine viruses kill 20 to 30% of the ocean's single-celled organisms every single day. Let that sink in for a second. Up to a third of the oceanic microbial biomass is literally wiped out daily by viral infections. But this isn't some sort of biological catastrophe. It's actually a highly functional engine. It's this massive daily turnover of cellular material that's fundamentally driving the way our planet breathes. Section 1. Marine viruses and carbon sinks, redefining the ocean's food web. So let's dive deep into the marine ecosystem to see exactly how this immense daily mortality rate fuels what scientists call the Earth's biological carbon pump. Now, for a long time, the impact of viruses on carbon export was heavily debated, but scientists have now nailed down two main pathways. First up, we have the viral shunt. When viruses burst a host cell, they release a flood of dissolved organic matter. This carbon gets shunted right back to local microbes who eat it, keeping it up in the surface layer. Then, we have the viral shuttle. The sticky compounds released during that cell lysis act like a biological glue. They clump organic matter and cellular debris together into what's known as marine snow, making it heavy enough to sink rapidly to the deep ocean floor, safely locking that carbon away. But here is where the research gets incredibly cool. What actually happens to the viral particles themselves after they infect a cell? Do they just float around forever? Well, in a cutting-edge experiment, researchers used nano-SIMS microscopy to literally track this. They labeled the carbon and nitrogen inside viruses with heavy isotopes, released them into coastal microbial communities, and watched what happened. And the resulting data shows the viral carbon and nitrogen actively entering the marine microbial community. The viruses aren't just bursting cells, they are literally becoming part of the food web. And these heat maps beautifully demonstrate a newly quantified biological process called Virivory. Yes, you heard that right, Virivory. Protists and bacteria actually eat the viruses. For the longest time, models assumed viruses were just removed by abiotic factors, like sunlight or sticking to particles. But these experiments proved that biological grazing is a massive factor. By actively consuming viral particles for their carbon and nitrogen, bacteria and protists increase the daily removal rate of oceanic viruses by up to 85% compared to just abiotic processes alone. That is huge. So, the crucial point here is how this elegantly structures the marine carbon cycle into a three-step dance. Step 1, viruses lyse or burst open their host cells, releasing carbon. Step 2, some of this carbon sinks via the fellow shuttle while the rest stays dissolved. And step 3, the viral particles themselves become a crucial carbon-rich meal for other microbes. They're recycling carbon back into the system directly through their own bodies. It's like a highly efficient microscopic recycling plant. Section 2, soil viruses fix carbon, the underground carbon hackers. Transitioning from the ocean to the land, we're finding equally massive viral impacts in terrestrial environments. Specifically, we're looking at intermittent flooded habitats, like agricultural paddy soils. And it turns out, soil viruses are nature's underground carbon hackers. In the soil, viruses use a totally different set of tools. They act as metabolic tuners, using what scientists call auxiliary metabolic genes, or AMGs. Now, these aren't the genes a virus needs to replicate its own physical structure. Instead, when a virus integrates into a host bacterium, it expresses these AMGs to fundamentally reprogram how that bacterial host processes and stores carbon. Think of it like a forced software update that makes the bacteria a way better carbon sink. And the genetic data from recent large-scale soil sampling backs this up beautifully. Across huge geographical stretches, researchers found that nearly a quarter, 23.7% to be exact, of all these auxiliary metabolic genes in paddy soil viruses are specifically coded to hack their host's carbon metabolism. For instance, they carry a massive amount of glycosyltransferase genes. These are the genes responsible for taking bioavailable, easily degradable carbohydrates and converting them into much more stable, complex carbohydrates. They are literally forcing the bacteria to lock carbon down into highly stable forms. Let's look at a really fascinating research experiment specifically tracking CO2 reduction in ecosystems by viruses. Researchers set up paddy soil environments under heavy metal stress. In one group, they added a concentrate of specific soil viruses, and in the other, they didn't. The results were absolutely staggering. Adding the viruses led to a 17.9% drop in CO2 emissions from the soil. At the exact same time, it caused a 35.4% increase in the long-term carbon stored inside the microbial biomass. By tuning the metabolism of their hosts, the viruses physically altered the carbon flux of the entire soil ecosystem. They directly reduced greenhouse gas emissions and boosted soil carbon storage. Section 3. The viral lifestyle switch. To burst or to integrate. All of this brings us to a really fascinating question. How do viruses actually know when to hack the metabolism and when to just burst the cell? Well, they essentially choose between two paths, and that choice is driven entirely by their environment. First, there's the lytic cycle, where they aggressively multiply inside the host, burst it open, and release nutrients. This cycle dominates in environments where carbon and resources are abundant. The alternative is the lysogenic cycle. Here, they integrate their DNA directly into the host's genome and express those AMGs we just talked about to help the host survive stress. This cycle takes over in low-carbon or heavily polluted soils. So, what actually flips the switch? It's environmental triggers. Researchers mapping paddy soils found that stressors like heavy metal pollution, which, by the way, is a major global change factor, actually increase the presence of carbon-fixing lysogenic phages by 10.7%. When the environment gets toxic or resources dry up, the viruses pivot. Instead of killing their hosts, they integrate with them. They protect and optimize the bacteria to ensure their shared survival. But when you return abundant nutrients to the soil, say when straw is returned to a field, boom, 93% of the viruses switch right back to the lytic cycle for rapid turnover. The crucial takeaway here is understanding this entire mechanism of sustainable carbon sequestration. It goes like this. Step 1. Under environmental stress, viruses integrate and trigger lysogeny. Step 2. They express AMGs to boost host carbon fixation and build complex, stable carbohydrates. Step 3. When conditions eventually improve and resources are abundant again, the viruses switch to the lytic cycle. They burst the hosts, releasing all those stable, complex nutrients, which then get converted into highly stable long-term soil carbon known as microbial necromass. It is just a brilliantly elegant, self-regulating system of carbon capture. Section 4. Shaping the global climate, harnessing the microscopic. Zooming back out to the macro level, we have to ask, what do all these microscopic viral interactions mean for our planetary climate models? Because human-driven global changes are actively altering these viral dynamics right now. In the oceans, studies using large mesocosms show that ocean acidification heavily alters phage-mediated bacterial mortality, especially in the winter. Meanwhile, eutrophication, nutrient runoff from human agriculture, consistently stimulates viral production across all seasons. On land, heavy metal accumulation in soils is actively driving genetic mutations in viruses, forcing a directional selection that favors the ones carrying soil carbon storage genes. We are fundamentally changing the environment, and the viruses are adapting, which ultimately dictates whether our ecosystems will store carbon or release it into the atmosphere. Today's explainer really shows us that viruses aren't just microscopic passengers on Earth. They are the invisible engineers of the global carbon cycle. We've seen the hard data on research experiments tracking CO2 reduction in ecosystems by viruses, reducing emissions in soils, and aggregating carbon in the oceans. So I want to leave you with this final provocative thought. As we map these microscopic metabolic tuners, could we eventually harness their power? Could we one day actively utilize viral genetic programming to draw down global CO2 and fight climate change? It's a fascinating frontier. Thanks for learning with me today. Keep asking questions and keep exploring.